Good morning. It's February 18th, and this is your daily brief in science. Here's everything you need to know. Researchers at Stanford have made a significant breakthrough in cancer genetics by identifying 380 key single nucleotide variants, or SNVs, associated with inherited cancer risk. This research, published in Nature Genetics on February 17, 2025, was led by former graduate student Laura Kelman and senior author Paul Kavari, MD, PhD. The identified variants mainly reside in regulatory regions of DNA, which influence the expression of cancer-associated genes, rather than in coding sequences like the well-known BRCA1 and BRCA2 mutations. To confirm their functional impact, the team utilized massively parallel reporter assays to test over 4,000 candidate variants, ultimately narrowing down to a functional subset. The study analyzed genetic data from millions diagnosed with the 13 most common cancer types, which account for over 90% of all human malignancies. Notably, the research uncovered a link between inflammation-related genes and cancer risk, indicating that interactions between cancer cells and the immune system may contribute to chronic inflammation that drives cancer. Gene editing experiments revealed that nearly half of the identified variants are essential for ongoing cancer growth. This lays the groundwork for future research into inherited cancer risk and potential therapies. The findings are expected to improve genetic screening tests over the next decade, facilitating more personalized risk assessments for complex diseases, including cancer. The researchers highlighted the importance of these variants in regulating biological pathways related to DNA repair, energy production, and cellular interactions, all of which are critical for cancer development. This study emphasizes the significance of inherited genetic changes over mutations that occur during a person's lifetime, marking a crucial step in the understanding of cancer genetics. While inherited mutations like BRCA1 and BRCA2 are already recognized, only a few are currently employed in risk prediction, indicating a pressing need for advancements in genetic screening. This research is part of a broader initiative funded by the National Human Genome Research Institute, aimed at mapping regulatory variants linked to various diseases, including cancers. A recent study has unveiled significant insights into how synthetic glucocorticoids impact brain development. Conducted by Leander Doney and colleagues at the Max Planck Institute of Psychiatry, the research found that prolonged exposure to these medications alters the balance of neuron types in brain organoids. The study revealed a concerning trend: an increase in inhibitory neurons coupled with a decrease in excitatory neurons. This shift creates an imbalance that has been associated with various psychiatric disorders. It suggests that the developing brain is more malleable than previously understood, with environmental factors, particularly glucocorticoids, playing a critical role in neuron type distribution. Fetal brains are especially sensitive to environmental influences, including medications taken during pregnancy, which can profoundly affect brain development. While the exact mechanisms remain unclear, it is evident that prenatal stress and medication exposure significantly impact this development. Collaboration with researchers from the Karolinska Institute in Stockholm and Helmholtz Munich further underscores the importance of considering both genetic and environmental factors in neurodevelopment and associated disease risks. Brain organoids derived from human stem cells serve as valuable models for studying early brain development and the effects of environmental exposures. The widespread prescription of synthetic glucocorticoids during pregnancy, affecting approximately 13 million newborns globally in 2020, highlights the urgent need for further research into their long-term implications on brain development and mental health. The authors of the study stress the necessity for additional investigation to understand the potential implications of these neuron imbalances for future mental health outcomes. While the research raises critical questions about whether these neuron imbalances increase vulnerability or resilience to psychiatric disorders, conclusive answers are still needed. Overall, the findings highlight the critical need to comprehend the long-term effects of altered neuron ratios on mental health. Recent research has uncovered an intriguing connection between our taste buds and heart health. At the 69th Biophysical Society annual meeting in Los Angeles, scientists revealed that the heart contains sweet taste receptors, similar to those found on the tongue. These receptors, identified as TAS1R2 and TAS1R3, were discovered on heart muscle cells and are functional, responding to sweet substances like aspartame. When these receptors are stimulated, they enhance the strength of heart contractions and improve calcium handling, which is essential for maintaining a healthy heartbeat. Micah Yoder from Loyola University Chicago noted that spikes in blood sugar after meals could activate these heart receptors, potentially affecting heart rate and blood pressure. The study also found that these receptors were more prevalent in patients with heart failure, indicating a possible link between receptor activity and cardiac disease. During heart failure, the heart may change its energy management, which could necessitate adjustments in nutrient sensing due to increased glucose usage. This research may provide insights into the correlation between high consumption of artificial sweeteners and irregular heartbeats, known as arrhythmogenesis. However, further studies are needed to explore the long-term effects of activating these receptors and their potential as therapeutic targets for heart failure. The stimulation of sweet taste receptors initiates a series of molecular events involving proteins that manage calcium flow and muscle contraction in heart cells. This groundbreaking finding highlights the complex role of these receptors in cardiac regulation and their implications for heart health. The increased levels of these receptors in heart failure patients underscore the necessity for further research into their role in heart disease, suggesting a novel pathway for understanding heart function and potential new avenues for treatment. Preterm birth is a significant public health concern, affecting approximately 1 in 10 babies and linked to higher neonatal mortality and morbidity rates. Defined as birth before 37 weeks of gestation, preterm birth often occurs without any identifiable cause, although factors such as low socioeconomic status, prior preterm deliveries, and certain health issues have been identified as risks. A recent study involving 50 pregnant women aimed to enhance early interventions for preterm births, which are associated with severe neonatal complications. The researchers focused on improving risk assessment by utilizing machine learning models that analyze routine blood tests and lifestyle questionnaires from the participants. Among the 5 machine learning algorithms evaluated, the Linear Support Vector Machine, or SVM, stood out as the best-performing model, achieving an accuracy of 82% in predicting preterm birth risk. Key predictors identified included C-reactive protein levels, hematocrit, platelet count, and maternal education level, emphasizing the multifactorial nature of preterm birth. The study revealed that lower educational attainment correlates with an increased risk of preterm birth, underscoring the importance of education in maternal health outcomes. While significant differences in delivery timing and clinical characteristics were noted between the study and control groups, no notable differences in age or body mass index were found. Despite these promising findings, the study acknowledges its small sample size and highlights the need for larger studies to validate the predictive models. The potential for machine learning models to enhance early intervention strategies, such as progesterone supplementation and cervical cerclage, could significantly reduce the incidence of preterm births. Data for this study was collected from Dr. Antoni Biziel University Hospital in Bydgoszcz, Poland, focusing on the medical history and health evaluations of the participants. A new era in vaccination may be on the horizon, as scientists at Stanford University have unveiled a painless topical living vaccine that utilizes the common skin bacterium Staphylococcus epidermidis. This innovative approach aims to enhance immune responses against diseases such as tetanus and diphtheria. Led by Dr. Michael Fischbach, the research challenges previous assumptions about the role of skin bacteria in immunity, revealing that Staphylococcus epidermidis can trigger a robust immune response. According to Fischbach, the immune system reacts preemptively to this bacterium, preparing the body to combat infections before they breach the skin barrier. At the core of this vaccine is a bacterial protein called AAP, which boosts immune recognition by displaying harmless fragments of tetanus and diphtheria toxins. In experiments, mice treated with the bioengineered Staphylococcus epidermidis showed significant antibody production and were able to survive lethal doses of these toxins, indicating the potential effectiveness of this vaccine in humans. This groundbreaking vaccine could eliminate the need for needles, making immunizations more accessible and affordable while also reducing common side effects associated with traditional vaccines. The study, published in the journal Nature on December 11, 2024, involved collaboration from multiple prestigious institutions and received funding from various health organizations and foundations. Looking ahead, human trials are expected to begin within the next 2 to 3 years, following successful tests on monkeys. Researchers aspire to extend this vaccine technology to combat a wide range of pathogens, potentially transforming the landscape of immunization. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.